Welcome to From Here Forward, a UBC Podcast Network podcast. I'm Carol, and she's Rumneek.
That's me. Hello. Today we're doing another one of our Changemaker episodes. This is where we profile different UBC faculty and alumni that are out there doing their amazing thing to change the world. Hence the title.
Now, I don't know about you, Rumneek, but I loved this Changemaker episode. I know academics get a bad rep sometimes for being nerds, but every now and then you do meet someone who's both smart and kind of funny. And you know me, Rumneek. I love it when men laugh at my jokes. That's all to say that in today's episode, Johal and I... Wait, do people know you as Johal?
Only you call me that, and people who don't know my name and address me that way in emails.
Cool. Okay. Well, anyway, Johal and I talked to, or rather learned from, Dr. Pieter Cullis from UBC about his revolutionary work with delivery systems and DNA and RNA-based drugs. His work was and is essential for the COVID-19 vaccines that we have today. A literal changemaker, if you ask me. Without further ado, here is that conversation.
My name is Rumneek, and somewhere on your screen is Carol.
Hello, good morning.
So why don't we just start off with you giving us a brief intro. Tell us who you are and what you do.
Sure. My name's Pieter Cullis. I'm a professor in the biochemistry department, biochemistry and molecular biology department at UBC. And for the last years, coming on 50 years, I've been working on... They're called delivery systems or nanomedicines, but it's basically systems that will take cancer drugs, for example, and just take them a little bit more specifically to tumors, to where you want them to go. And for the last 25 years, I've also been working on encapsulating nucleic acid-based drugs, DNA and RNA-based drugs. So this has resulted in, we've had over the years, five drugs approved by the FDA and other regulatory agencies. 3 of those to do with cancer and its complications, and 2 that are using the nucleic acid-based drugs. One of them to cure a rare disease known as transthyretin-induced amyloidosis, which is a bit of a mouthful. And the other one is something that people are quite familiar with, which is the Pfizer-BioNTech COVID-19 vaccine, where we developed the lipid nanoparticle that encapsulates the messenger RNA and allows it to get inside target cells. Without the delivery system, the mRNA vaccines wouldn't work.
So we heard that you started at UBC around like 1964, which my math says is like 60 years almost. So what has it been like to know that you've spent 6 decades at one institution?
It's a pretty good feeling. I enrolled in undergraduate physics in 1964 and graduated with a PhD in 1972. And then from 73 to 77, I spent 3 years in Oxford in the UK doing postdoctoral work, research. And then subsequently in Holland, I was there for a year as a postdoc. And then I came back in 1978 to a position in the biochemistry department. So it's been a long-term association, that's for sure. It's been a very rewarding one in many ways. I mean, people can go to different institutions, but it's really good when you're in a place that where you what's the word? The resources are there. Obviously, family plays a role as well. And as a result of that, what I've managed to do is not only, from an academic point of view, have a fairly successful career, but also I played a big role in doing a lot of startup companies. I've done 12 startups, which now employ about 400 to 500 people in the Vancouver area. You know, all of my students, or many of my students, would end up in the U.S. to one company or institution or another. But I said, we got to stop that. And the only way I could figure out to stop it was to start a company where we employed people that were in my group. And we kept them local, and that has grown into, as I said, we're now the global hub for these lipid nanoparticles, which is having a huge impact worldwide. but now the world is coming to us to use this technology, not just for vaccines, but for many other pharmaceuticals, for many other drugs. And so it's a very exciting time, but it's been vital to have been here over a long-term basis. And UBC has played a huge role in that.
Wow, that quite the trajectory. Carol, I feel like we need to go back to school because 12 startups and all that seems really, really impressive. So thank you for kind of tracing that. So your revolutionary work through the years have helped make mRNA COVID vaccines possible. So we're very unscientific people, Carol and I. So could you please kind of explain, how that came to be?
Sure, and a part of that has to do with some of the startups, some of the companies that we've co-founded, because it's really been very much of a, joint endeavor, a collaboration with between UBC and my lab and these companies. But way back in 1985, as I was saying, I had a very good team in my lab, and we developed an instrument to make lipid nanoparticles that it turned out other people wanted. I gave a talk in New York at one time, and one of the people in the audience called me up after and said, of course, in those days there was no internet or anything, and said, I want one of those instruments. So I said, well, fine. And I gave him the contact information for a machinist who had made the apparatus for us. Well, a couple of months later, I got this very irate phone call from this scientist in New York saying, "Well, I got this thing, but it doesn't work." And of course, the machinist had no idea what the apparatus was used for. It was called the extruder. That's what we called it later on. And there was no quality control, or could it actually make the nanoparticles? And so that's when we decided, okay, there's an opportunity to start a little company, where we would get the machinists to make it, we'd quality test it and make sure the thing worked, and then ship it out. And so this was in one of the people in my lab. It would be in his garage that we assembled these things, and we sold hundreds over the years at a couple of $1,000s each. And the this funded parties and other things. It was really a lot of fun. And so that was the beginning of our entrepreneurial efforts. Somewhat later, we started to work with a company in Princeton, in New Jersey, that was called the Liposome Company. And again, I wanted to keep the team together here, and so I said, okay, in order to keep on working with this company in Princeton, they needed to form a subsidiary here in Vancouver, so that we could keep the team in the same place, which is what they did. And so for 5 years, we formed the Canadian Liposome Company, as it was termed. And this is all leading up to the vaccine in the end. After 5 years, the new CEO came in and basically said, well, you should all move down to Princeton. And he offered us high salaries and all the rest of it. We said no. And and started their own company at that point called INEX, INEX Pharmaceuticals. This was about 1992. And we were working on delivering cancer drugs using lipid nanoparticles. But in 1995, the CEO that I'd hired, he came to me and he said, "Well, I really can't raise money for the company. Putting these old cancer drugs in liposomes. We need to be doing gene therapy." And he felt that had a lot more appeal to investors. And so then we had to switch to that, to these trying to get these big molecules. They're at least in terms of drugs, they're big molecules. Trying to get these big molecules into cells in the body, and that was work that we, for about 5 or 6 years, we worked on trying to get systems that were not too toxic, because a lot of the ways of introducing DNA or RNA can be quite toxic. Anyway, so in about 2005, we were contacted by a company in Boston called Alnylam Pharmaceuticals, And they said, we want to deliver something called small interfering RNA, a piece of RNA that will very specifically, it causes a particular protein not to be made. It silences the expression of that particular protein. And so we worked with them for about 7 years, which resulted in a drug that went into the clinic in 2012 that was to treat the disorder that I mentioned a little while ago, transthyretin-induced amyloidosis. That was very successful. But in 2012, another company that I co-founded called Acuitas, we decided, okay, if we can deliver this small interfering RNA or siRNA, maybe we can deliver a messenger RNA in the same lipid nanoparticles. And that turned out to be the case. And so we were trying to express these proteins in the liver so that be an intravenous injection, and then we would be getting proteins made in the liver. And those proteins that were made were coded for by the messenger RNA that we were putting into the lipid nanoparticles. So with all kinds of diseases, I mean, a child is born with, some hereditary disease. They're not making a particular protein. Maybe it's involved in blood clotting, so they're hemophiliac, or maybe it's Tay-Sachs. There's many disorders. These rare diseases, and so the notion was and still is that we can get the liver to make those proteins, and so therefore rescue the child. But then about 2014, we got contacted by a scientist at the University of Pennsylvania, a guy named Drew Weissman, and he's an immunologist. And he said, "I really want to try your system as a vaccine." And so instead of having messenger RNA coding for a protein that was being made in the liver, uh, in this case, the encapsulating messenger RNA coding for a protein that was associated with a virus. In his case, he said, "Okay, let's try Zika virus." And so we put the messenger RNA in, coding for a surface protein on the Zika virus, and sent it to him, and it worked extraordinarily well. And so that resulted in a collaboration between a company in Germany called BioNTech and Acuitas that started about 2017, 2018. And the objective there was to develop a vaccine for influenza. Now, it turned out that BioNTech was also working with Pfizer on an influenza vaccine. And so in January, February of 2020, when the pandemic hit, all efforts were switched, of course, to making a vaccine for COVID-19. And so the lipid nanoparticle that we'd been supplying for the influenza virus was moved over to encapsulate the messenger RNA for the spike protein on the SARS-CoV-2 virus, which is the virus that causes COVID-19. And Pfizer-BioNTech took that into the clinic, and of course the rest is history. In November 2020, the results were announced that this vaccine was 95% effective for preventing infection or preventing COVID-19, and across all ages and ethnic groups, etc. And so I think last year there was over 3 billion doses delivered, and this year it's a heck of a lot of it too. I think 4 billion people in the world have been felt the benefit of this vaccine. So it's had a huge impact that one could never prophesied based on what we were doing, say about 3 or 4 years ago. So it's been a great surprise to us, as you can imagine, because we've been working to show that these lipid nanoparticles can be important delivery systems, but to have it kind of validated or proven in response to a global pandemic is really quite a startling situation. Anyway, so it's really changed this is now an established therapy for vaccines, but even more exciting is the fact that the same approach can be used to treat really any disease you care to name, ranging from cancer to heart disease to neurological disorders, Alzheimer's, etc. And so it's a very exciting time in medicine as we apply the nucleic acid therapeutics using these lipid nanoparticle delivery systems to treat, a huge range of disorders. I hope that's not too scientific.
You brought us along, and I see the dots. So I just kind of want to go back to something that you said a few times is that you wanted to have your lab kind of local and keep people with you as opposed to them leaving. Why was that so important for you to maintain that locality with your lab and your researchers?
As Canadians, we've got to be concerned about the fact that, I think 40% of our science, technology, engineering, mathematics the STEM graduates, as it's termed, go south. They will take jobs in the US. And so, what are we doing? Training people, from the Canadian taxpayer point of view, this is not very good. Training people that then benefit, say, the US or other countries. That's certainly one driver for me. But the other is, if you've got a core of well trained, highly motivated people who get along together, then and then you can really build on that in a company environment. If you get that kind of team operating, as you can see from what we've done, you build on that expertise, and then you see that the thing is, it's not just the academic or the discovery side of it. You also build say, the intellectual property portfolio, so that you then have, for example, we have many patents through Acuitas and the other companies on the components of the lipid nanoparticle, the ones that work the best. We've been so that's a that, so Acuitas can license that to other companies. You build a lot of value in the companies locally, and of course, that has huge benefits in terms of the ability to. Right now, we're starting up, companies to go after blood disorders, to go after neurological disorders, skin disorders, you name it.
You touched briefly on the connection with Pfizer. Can you explain further how the connection with Pfizer and Moderna came about and kind of did they approach you? Like, how did that come to be? And what can you tell us about that?
Say with Moderna, we actually through Acuitas, we were working with Moderna. This was in the, I think, 2013, 14, 15 time period. But then Moderna decided that they felt they had intellectual property that allowed them to move forward with their own lipid nanoparticle. This was really the technology was really something we developed with Alnylam, and the lipid nanoparticle used there for the drug for transferrin-induced amyloidosis. This was the siRNA. Anyway, so some of the ideas were there, and so as a result of that, Moderna felt that, they developed their own proprietary version, and so that relationship stopped somewhere in around about 2016, as I remember. But then with Pfizer-BioNTech, as I mentioned, we were working with BioNTech on a flu vaccine, and BioNTech in turn was working with Pfizer, and so that's how that relationship evolved. Actually, it was kind of funny. In February, towards the end of February of 2020, I was invited to give a talk in Pearl, it's called Pearl River, which is just north of New York, and it was the Pfizer headquarters. And I couldn't really understand it. Usually, when I give talks in, say, pharmaceutical companies, then it's basically to the scientists that I'm talking. It's not really to the business people or some of the people higher up in the organization. But in this talk, there was vice presidents. They all wanted to listen to what I was talking about, and I said I couldn't quite understand that. It turned out so that they were just then making the decision as to whether or not to go forward with the lipid nanoparticle mRNA vaccine. And as you know, they made a heck of a bet there, because not only did they start the clinical trial, which started in March or April of 2020, but they also started scaling up and anticipating that the clinical trial would be successful. So, in other words, if the clinical trial results were not good, then they would have basically wasted, $500 million or some staggering amount of money. And so it was a time that the whole company, it's a huge company, but was really focused on whether or not they should go forward with the mRNA vaccine, which was, a really relatively unproven technology at that point. It's not a classical approach to making vaccines. Obviously, the preclinical results were astonishingly good, but was this really going to work in people? That was still a question. Yeah. Anyway, so they obviously made the right decision. But I think the CEO more or less bet his job on that. The If that clinical trial hadn't worked out, I don't think he would be CEO anymore. He took a chance, and it really had a big impact. It's a guy named Albert Bourla.
I just kind of want to go back to something again that you said about how like this wasn't really something that you anticipated in the success and how many people would be taking part in this technology. Like, what is it like knowing that so many people have benefited from your work? Like, or is it something that you're just like, cool, it is what it is, or is it every day where you're like, wow, that's so wild?
Well, it's amazing. It's absolutely amazing, right? I mean, who would have thought? Because, as I said, 3 years ago, nobody knew what a lipid nanoparticle was, and while we thought this stuff was pretty important, we certainly didn't have the rest of the world convinced of that. And then suddenly, we have a global pandemic, and to be playing a bit of a role in that is amazing. But really, I just feel very happy that something that I started way back in the 1980s with about 4 postdocs in my lab, has had such an impact. So you feel pretty good about that. So occasionally the four of us will go, or a few of us will go out for a drink and celebrate that. It's still, none of us can really get our heads around the fact of the big impact it's really had. It's quite amazing. Yeah, obviously, a lot of that is just plain luck, or whatever you want to call it. We were in the right place at the right time. If the pandemic had happened a couple of years earlier, there was a bunch of things that came together, such as the messenger RNA I was mentioning. Drew Weissman at the University of Pennsylvania, he wouldn't have been they wouldn't have been ready for that side of the equation, and we wouldn't have been ready from the point of view of the delivery system. So yeah, a lot of things had to come together that you couldn't forecast. It points out a lot of the non-linear nature of science, right? I mean, we didn't start out to make a vaccine at all. We were trying to make something that made proteins in the liver, and then suddenly you get called up, and let's try it as a vaccine. Okay, but my knowledge of immunology is very scanty. As I say, it turned out that it worked extremely well. Anyway, so these are the kind of events that can happen if you get involved in something and push it as far as you can, and then things you don't expect can occur. I always encourage people to take chances a bit because like, for example, my training is in physics. I got a PhD in physics. I said, okay, I didn't want to do physics anymore. That things in the life sciences looked way more interesting. So I took a bit of a chance there. After a couple of years, I found I could maneuver in that environment. And then I thought, okay, well, I don't know anything about drugs, but let's have a go at that. Or don't know anything about companies, but let's have a go at that. And I think that's, you gotta, when you move out of your comfort zone, as it were, and do things that you're not exactly all that confident you can do, then interesting things can happen that are obviously in my case have worked quite well. So there's been some downsides as well. I'm not saying it's all been totally linear, but it makes for an interesting life. Let's put it that way.
That is great advice, Carol. I hope you're taking notes because we need to, I mean, not that we're going to come up with some great invention that's going to save billions of people's lives, but maybe something close. So to backtrack a little bit, you talked about how you spent most of your career at UBC and again, giving back to the local community in terms of providing jobs for people here was very important to you. So can you tell us a bit about how UBC contributed to your success and kind of if you see it as unique and special as an institution, what do you feel that's so unique and special about it that differentiates it from other institutions that you've perhaps spent time at?
Oh yeah, yeah. No, I mean it's had a huge impact, and one of the things that's been good about what UBC has done, as I mentioned, I came back here in 1978 as an assistant prof in the biochemistry department, but the opportunity came along to, as I say, start a company, which we did in 1985, I guess. This is a Canadian liposome company, and so I went to the to my head of department and said, "Was it okay if I go on half time?" I was working half time at the university and then half time at the company. And with the course being that I'd only get paid for half time. He was quite enthusiastic about this because it helped the finances of the department. But anyway, so that was the kind of the flexibility that UBC gave me to be able to start up these startup companies, and of course also assistance with the intellectual property. So things that were invented in my lab, then the university would so certainly helped in terms of patenting, writing the invention disclosures and the patent applications, and making sure that that side of things was taken care of. And then helping quite directly in some cases with, if we wanted to develop something in my lab, then facilitating that process so that we could actually take it to a point where it was maybe something could actually be commercialized. In the case of some of the instruments we worked on. Anyway, so it's been a very productive relationship in that way. So it's yeah the that's been a huge benefit that UBC has been, say, so flexible. It's also just an amazing resource in terms of, when you got a problem, then there's people in the university who can help.
I mean, most of this conversation has just been the fact that you've been very, very successful in your life, and you've kind of hinted at certain things, but um, what is an advice or perhaps a secret that you have gained throughout your life that has taught you about success and how to be successful?
Well, it really comes down to moving out of your comfort zone and doing something. I mean, very often these are things you want to do, but you sometimes feel held back because you don't know what to do. So an example. I mean, just getting into anti-cancer drugs, which we did in the 80s. The We didn't know anything about how to develop a cancer drug or anything like that. But it's surprising, if you go and chat with people. In this case, we talked with an investigator at University of Toronto and said, "Look, we've got this. It was a liposome. It was a lipid nanoparticle with a cancer drug in it, and the drug is, it's a drug called doxorubicin, which is a very common anti-cancer drug, but it has a very nasty toxicity that it can cause your heart to it causes cardiotoxicity. It causes the toxicity to the heart, and it can cause heart failure. And so, obviously, it's not very good if you get cancer and you're being treated for cancer, and then the drug can cause your heart to fail. Anyway, so it turned out that with this delivery technology, these lipid nanoparticles, they weren't accumulated by the heart, but on the other hand, they did tend to accumulate in the tumor. And so we could have a less toxic version of this cancer drug. And so we went and presented those results to a clinician, and who would do phase one trials, as they're termed. I mean, initial trials in people. And yeah, he said, "Sure, I'll take that into the clinic." And so something where we didn't have any idea of how to do things, but if you reach out enough, then something happened, and that drug ended up getting approved for metastatic breast cancer some 10 years later. So by moving outside your comfort zone, as it were, and then trying to make contact with people who might be able to help you to go to that next level. Can't say it's been a particular linear path, but just by virtue of starting something off, it's amazing. It's amazing what happens. So in terms of advice, I mean that's probably the biggest one is to take a chance every now and then. If you really want to do something, even though you may not have a clue as to how to do it, well, have a go. Surprising things can happen. You're going to have some fun doing it. You always bring things, when you change fields a bit, you always bring something to the new field that other people don't have.
And so, just to kind of wrap up, although we may, I don't know, Carol sometimes surprises us with the last question. So I'm going to ask my last question. And Carol, I don't know what you're going to do after that. But scientists have alluded to potentially more pandemics happening in the future. I mean, obviously, I don't know if you can predict the future, but what does the future hold in store with, in your opinion, with the advancement of drug deliveries or this kind of drug delivery? And do you have any kind of thoughts as to where this is going?
I often refer to what's happening as being the third generation of pharmaceuticals. I mean, first generation drugs are these small molecule drugs like cancer drugs or aspirin, or basically they go everywhere in your body, and they obviously have some benefits, but they can have some downsides too. The second generation drugs are what you might call biologics, or like antibodies. There's certain antibodies that are used for treating cancer and other diseases. But these third generation nucleic acid based drugs, or they're a form of gene therapy, really have an enormous range of applications, not just as vaccines. And so pretty much any disease you care to name, you can treat using an mRNA based therapeutic. And also, it just changes the equation. As an example, the say with sickle cell anemia, which is a pretty prevalent disorder, genetic disorder, that requires us to get our lipid nanoparticles with messenger RNA to bone marrow where the blood cells are made. Well, we're just starting to be able to do that. We're very good at doing the liver, but anyway. So, but as soon as we're able to do that, able to deliver mRNA that say codes for production of proteins that will affect which form of hemoglobin is made, then you can easily see a cure for sickle cell anemia emerging, or cystic fibrosis if you can get to the lung. And there it's a protein called the CFTR. It's an ion transport protein. If we can get that corrected, then that disorder will be rectified. Heart disease, there's now high levels of high density lipoprotein, or low density lipoprotein, sorry, the so-called bad cholesterol, leads to heart disease. Well, now you can find ways of turning that on and off, or modulating the levels in the circulation, again using these messenger RNA-based techniques. And so it's a very, I mean, in the brain we can we can transfect certain tissues in the brain, and neurological disorders become open territory. So you're going to see really in the next 10 years, I think an enormous number of new drugs coming along that are based on this mRNA approach. The other part of this that people haven't really recognized is that it's so fast, because once you know what protein you want to stop making or start making, or you want to edit, then making the messenger RNA that will perform that function, will take maybe, say a month or so, maybe a little bit longer, but not much. Packaging it up takes a day or so. And so then you have this highly targeted drug that's to perform whatever function you want. And so it's much cheaper and much faster than our current way of making drugs, which it can be 10 to 15 years and $1 billion. Well, now we're into a month or two, and maybe it's $200,000 or $300,000. So it's a very different equation, and it's going to revolutionize medicine, from what I can see.
So I've heard a lot of successful people have a routine or a thing that they've done consistently throughout their lives, whether it's drinking coffee black or waking up at 5 a.m.? What is something that has been consistent in your life that you've done for a while?
I suppose one thing has been that I've always been part of teams, whether it's, teams in my lab or teams in these companies. And I really enjoy that, working in an environment where I'm working with a bunch of other people and we have a common aim. So as long as I keep that going, then I'm going to keep on going as long as I can. As I try to emphasize that without UBC, none of this would have happened. So it's been certainly UBC has been very supportive over the years, and it's been a good relationship. That's for sure.
Well, that was a really great conversation. And as Cullis said, big shout out to UBC. She's really doing her thing and changing the world.
Definitely. I mean, I thought I wouldn't have understood a single thing during that conversation, but I feel like I learned a lot and it made a lot of sense. And the potential for Cullis's discovery to treat other diseases, including cancer, heart disease, and Alzheimer's, is something that cannot be understated.
And I do feel like we get to talk to a lot of really cool people as journalists, but I've got to say, Dr. Cullis was probably the most coolest so far, in my very objective opinion.
Yeah, I mean, the fact that he contributed to something that was so pivotal in our lifetime and honestly likely changed the course of the pandemic and saved lives is wild.
I know I said this last time, but I feel like a little luck and a lot of hard work does go a long way. Personally, though, I wish I had more luck in my life, but here we are.
I mean, just stay hopeful because, as Cullis said, if you keep trying, you never know what could happen, Carol.
That is a wrap from us, your favorite hosts ever.
Thanks, everyone, for listening. Make sure you catch our next episode by subscribing or following our show on Spotify, Apple, or wherever you get your podcasts. And if you're feeling your feels, please drop us a review. You can also find me on Twitter at Rumneek with 3 E's.
And me at Carol Eugene Park.
Today's episode was recorded at CITR Radio and engineered by Hina Imam. From Here Forward is an alumni UBC podcast produced by Podium Podcast Company.